All right, in chapter 8.4, we're going to discuss the regulation of gene expression. So we talked previously about how gene expression occurs. We have transcription and then we have translation, but what controls that? That's what gene regulation means. What controls it? We're going to talk about some of the reasons why a cell might need to regulate its gene expression. We'll talk a little bit briefly about the levels of gene regulation and explain why it's useful to regulate gene expression at different levels. And then we'll talk about some regulator proteins that interact with chemical signals to change the rate of gene expression. So in a bacterial cell, there are some proteins that are needed all the time. Something like RNA polymerase is needed all the time because it's required to make new proteins. But other proteins might only be needed at certain times, like if you need to make proteins to break down lactose, right? Lactose is a sugar, but lactose isn't always present. So the question is, why would you make enzymes to break down lactose and waste all that energy when there's no lactose around? When lactose is present, you probably want to make the genes to break down lactose. So this is an example of how microbes can respond to their environment. Environmental response is the biggest thing that leads to gene regulation. So the temperature changes, there's new food available or no food available. That's when microbes are going to change what genes are being made. To compete successfully, they can't waste energy, right? They can't make unneeded proteins. Every protein they make spends energy, so they can't waste that if they don't need those proteins. So that's a big reason that microbes need gene regulation. And again, that's a reason you need gene regulation. Also, as you grow and develop, different genes are needed at different times. So if we go to the simplest level, there's kind of three places that gene regulation can occur for us. This is a super simplified view. Gene regulation is very complicated in reality, but we can have transcriptional control, so regulating when the DNA gets transcribed into RNA. We can control translation, how much translation is occurring. So yeah, RNA maybe got made, but whether it gets made into protein or not. And then we can do what's called post-translational control. This is modifying proteins in different ways. You can add little switches to proteins that can turn them on and off. We're going to look at some very simple examples of gene regulation. We're going to focus on transcriptional control, and to do this, we need to talk about some proteins that can bind to DNA, so they can grab on to a sequence of DNA. So usually we have a gene, right? And it's got a promoter. This promoter might have what's called regulatory regions or sequences in it. These regulatory sequences are regions where proteins can bind to. These proteins might either turn on transcription, we would call that induction or upregulation, or the proteins, if they bind, they might turn off gene expression. They might be repressors or downregulators. It really depends on the sequence and the region that we're talking about. These regulatory proteins are encoded by yet other genes, so it becomes very complicated very quickly. So we're interested in this purple gene. Let's say that this protein, when it binds to this regulatory sequence, it blocks RNA polymerase. So if that's the case, the regulatory protein binds and RNA polymerase cannot transcribe the target gene. When this leaves, then RNA polymerase can come in and transcribe. Microbes are often sensing different conditions, and the proteins that are binding to either block or recruit RNA polymerase are generally sensing things in the environment. Usually they'll bind to small molecules we call ligands. In this example, this is diphtheria. So diphtheria is a pathogen that needs to make a toxin. It needs to make this toxin because it's trying to extract iron from the cell. When there's high iron around, diphtheria does not need to make the toxin. This toxin kills cells which release iron. This repressor protein, as it's called, binds to the regulatory region and actually blocks RNA polymerase from expressing this gene. When iron levels drop, when there's no iron around, the repressor protein falls off. It changes shape and it gets moved. That is removing the repressor protein, which turns on transcription. And then diphtheria now makes the toxin gene, which goes out and kills host cells, which release iron, which in turn will then, if there's high iron, will lead to the repressor coming back because we no longer need that. So we're going to look at the most famous example of gene regulation. This is called the lac operon of E. coli. The lac operon is a series of genes that are required to break down the sugar lactose. LAC-Z, LAC-Y, and LAC-A are all genes that encode proteins that help E. coli break down lactose. LAC-Z is the gene, the protein that actually does the breaking down of lactose. Lactose permease, LAC-Y, helps it get into the cell. And LAC-A, we don't fully understand its role in the process. We're still learning stuff about this, even though we've been studying it for over 70 years at this point. So these genes are all needed to break down lactose. There's a promoter region here with a sequence in it called LAC-O. This is not a gene, but instead a region where something binds. We also have another protein called LAC-I. LAC-I is a repressor. This is not part of the lac operon, it's made separately. The LAC-I repressor normally binds to the LAC-O sequence here. It binds to and prevents the making of this when there is no lactose present. So in most cases, lactose is not present, so the repressor protein binds here, and we don't make LAC-Z, Y, or A. We don't need them, right? There's no lactose. Why make the genes to break down lactose? So with this LAC-I repressor bound, no transcription occurs. All right, now here's where the sensing comes in. Lactose molecules, the sugar lactose, when it enters the cell, it binds to the repressor molecule, LAC-I, and it actually changes its shape and causes it to fall off the DNA. This opens up the promoter, and now RNA polymerase can come in and transcribe the genes, making LAC-Z, Y, and A. Because there's lactose around, we need to make the genes to break down lactose. So, the two key points here, right? When there's no lactose, the repressor binds and prevents the transcription of these genes. When there is lactose present, the repressor changes shape, it senses that the lactose is around, and then RNA polymerase can come in and make LAC-Z, Y, and A. It's a very elegant and beautiful system, right? It's the way that bacteria can sense what's going on in their environment. We've talked about this on several different occasions when we talk about differential plates, right? We talk about colonies that can ferment lactose versus those that cannot. This lac operon is a key part of those bacteria. Okay, so this topic is huge. This is one of my favorite things to talk about in genetics, but of course we're going to keep it just this simple. So microbes at their core need to sense the environment, right? And in turn regulate gene expression to optimize their growth and survival. Why make a gene to break something down that is not there, right? That's the key message I keep reiterating. We talked about transcriptional control, which regulates whether RNA polymerase can come in and transcribe a given gene or not. We have activators. Those are things that increase the rate of transcription, and repressors, like we saw in the lac operon, which decrease the rate of transcription. A lot of times we have signaling molecules that can bind to repressors and pull them off of the DNA, allowing transcription to occur, just like in the lac operon example. There are, of course, additional ways to control gene regulation. How tightly wound or unwound the DNA is. You can regulate translation. And then, of course, post-translational modifications. You can adjust proteins in different ways. And even breaking down proteins, right? If you get rid of an enzyme, that's a form of control. All right, that's it for 8.4.